In our last video, we're going to continue talking about the effects of multiple factors on a given phenotype. So we're going to start out talking about multi-gene traits. So this is different from multiple alleles, because this is actually many different genes and of course their corresponding alleles, all working together to produce continuous variation. So this is something called polygenic inheritance. So polygenic inheritance results from polygenic traits, which are those phenotypes that are influenced by the input from many different genes. Now, this is often 3, 4, 5, even 80, 100 genes that are all working together for a particular trait. Now, the characteristic of these polygenic traits are continuous variation. So a perfect example of this is height. If you were to line up all of the individuals on campus right at this moment, all of those individuals would fall into a range of height. There would not be sort of a small, medium, large height category. We could randomly assign categories, but if you were to line everybody up in height, you would see that there are no distinct categories. And this is because there are at least 180 gene locations that influence adult height. Another perfect example of this is skin color. So you can see that picture of the ladies down below. We have everything from super light skin color all the way to really dark skin color, and then everything in between. And this, again, is due to many different genes all working on the same trait. Now, whether we're talking about hair color or even eye color, height, skin color, even to some extent weight or the general build of your body. These are sort of all additive effects that contribute to the phenotype. So an individual who is shorter is going to have more short genes and less tall genes. An individual who's taller is going to have sort of an accumulation of taller genes. Same thing with skin color. Individuals who have darker skin color have more genes that are encoding darker pigment or affect the darker pigment. Individuals who are lighter in color are going to have less of those dark pigment genes, if they have any at all. This individual here might not have any dark pigment genes. Very often, the sort of variation that we see in the human population is due to these polygenic traits, and a lot of the characteristics that we see as varying among a large group of individuals or even among distinct populations are due to multiple genes all working together. So what's interesting about this is there are behavioral traits that are also polygenic. So many behavioral traits are influenced by the effects of multiple genes. So an example of this is autism. So autism appears to be the result of maybe 10, maybe 20 genes that are all contributing or interacting to produce this phenotype, which is autism. So individuals with autism are characterized by, or the disorder is characterized by difficulty interacting with others, particularly in making emotional connections. They tend to have narrowly focused interests, repetitive interests, but autism is pretty hard to study, and that's just because the symptoms are regulated by many different genes. They're also probably somewhat affected by the environment. And so when you have multiple genes that are involved in a particular trait, it's really hard to isolate the effects of any individual gene, because any individual gene might have a very small impact, but altogether with all 20 of those genes or all 30 of those genes, however many there are, they sort of have this accumulative effect. So genes responsible for autism may also influence some desirable characteristics. So this comes from the idea called the geek theory of autism. And this came from researchers who documented an overrepresentation of autism among children whose parents work in engineering, physics, computer science, and math. Now, so the idea here is that maybe these individuals who go into these have particular traits, particular characteristics that make them good in these fields, and then when you sort of get too many of them, too many of these traits, it could possibly lead to autism. Now, I don't know how supported this is. It's just sort of an interesting theory. This would probably fall under the general use of theory, where just a sort of I have a theory thing rather than something that's been supported and is our sort of scientific definition of a theory. But in any case, it's a good example of many genes affecting the behavior of a particular individual. We can also have the opposite. So instead of many genes affecting one trait, we can also have one gene that influences multiple traits. So a really good example of this is sickle cell. I like that our book keeps going back to some of these particular disorders and sort of tying them into each unit. So we saw sickle cell in lab. We saw sickle cell when we were looking at changes in gene expression and the effects of changing an amino acid and producing a different protein. So let's look at this with respect to an actual phenotype. So we know that there's one gene that influences sickle cell, but it has many effects. Now this is called pleiotropy. So pleiotropy means that there is one gene, but there are many seemingly unrelated phenotypic effects. So remember that if you are homozygous for hemoglobin gene, you have these nice round plump normal hemoglobins. There are no sickled cells, and so you don't have any disease, and your blood is normal, like this guy here, and can carry oxygen regularly. If you are heterozygous, you produce both. So we saw this in our lab when we saw the individuals who are heterozygous. They produce some sickle and some normal hemoglobin. And so here we've got our sickle shape blood. And then we have the homozygous individuals who have the sickle cell blood. Now remember that sickle cell is potentially fatal. Individuals that produce these defective red blood cells have a decreased oxygen carrying capacity. These defective blood cells accumulate in blood vessels, cause extreme pain. So individuals with sickle cell often suffer from shortness of breath and a bunch of other problems that are related to the inability of your body to move oxygen, and then of course the accumulation of these sickled cells. So there's our pleiotropy. One thing that is not particularly relevant here with respect to pleiotropy, but is really important to the study of sickle cell is that having the sickle cell trait or the heterozygous situation allows the individual to be immune to malaria. Being homozygous for sickle cell also does, because the virus that is it's a bacteria that is moved by mosquitoes can't affect those sickled cell blood cells. So you end up with sort of an advantage of being a heterozygote in a situation where malaria is present, because you're not going to die from malaria because your blood cells can't be infected, and you're also not going to die from sickle cell. So I guess in a way this is sort of pleiotropic because the gene for sickle cell does give you sort of an immunity to a different disease on top of the other characteristics of the disorder. All right, so let's switch gears a little bit. This is kind of a big flip. And let's talk about sex-linked traits. So recall that we said that there are two sex chromosomes, an X and a Y. Mom only passes on an X. Dad can pass on an X or a Y. Now also recall that the Y chromosome is kind of special in that it only encodes genes for the development of male sexual characteristics. Now that means that if you are looking at situation comparing an X and a Y, that there are genes on the X that are not present on the Y and vice versa. Now this leads to a really interesting pattern of inheritance called a sex-linked pattern of inheritance. And it's why we see some disorders more frequently in males than in females. So, color blindness occurs on the X chromosome. Now, if you've ever met anybody who is colorblind, you probably have met a male. It's very rare for a female to be colorblind, although she can be. So if we looked at the X chromosome, we have this R gene. It codes for a specific light-sensitive protein that gathers particular wavelengths of light and allows you to see color. The other version, a recessive allele, is defective, which means the individual is colorblind. I believe this one that they're talking about specifically is red-green color blindness, which is also the most common colorblind. Now, normally, if you are going to have a recessive disorder, you need to get two recessive copies. So that's what's going on here with this female. She has two recessive copies, and so she would be colorblind. So she has inherited this disease just like anybody else would inherit a recessive trait. However, if you are male, you only get one X chromosome. Your Y chromosome, because it only encodes that SRY region, the sex determining region, doesn't have a homologous allele for colorblindness. So if dad gets a this guy gets a colorblind allele from his mom and a Y chromosome from his dad, he's only getting one copy of that colorblind allele. So that means the only option for him, even though he has only one allele, is to be colorblind because his one allele is the dysfunctional light-sensitive protein. I hope that makes a little bit of sense. I can answer more questions about this in class if you want. So, in order for a male to have normal vision, he has to get a normal allele from mom because he's not getting any allele for this trait on the Y chromosome. Females, of course, because they have two copies of the X chromosome that behave just like the other autosomes, can either be heterozygous or homozygous for normal. So when you think about the female, she's actually occurring, her color blindness is actually occurring along sort of normal patterns of inheritance, whereas the male is only getting one recessive copy. Now, one recessive copy normally means that you don't see the trait, but here, because he only has one, that's the trait that we see. So when we look at pedigrees, we actually find that in a pedigree, if you have a disproportionate number of males, you probably have an X-linked disorder. Because otherwise, males and females are affected equally. They're subject to the same probabilities as any other inheriting any other recessive allele. So by when you look at a pedigree, one of the first things that I like to do is look to see, are males affected much more often. So usually in a large pedigree, if you have an X-linked disorder, you're going to have lots of males and little to no females that are affected. So normally, the pattern of inheritance is the same whether you are male or female. So you have an equally likely chance of inheriting either allele from your parents. It has nothing to do with whether you are male or female. But if that gene is encoded on the X chromosome, then you are going to see a different pattern of inheritance between males and females. So another example of this is hemophilia. So that disease was very prominent in European royal families in the late 1800s, because there was a lot of interbreeding, and I shouldn't say interbreeding, intermarriage between the royal families. So it was believed that, I actually mean early 1900s, Queen Victoria was actually thought to be a carrier, meaning that she had a recessive hemophilia allele on one of her X chromosomes. And if you Google hemophilia pedigree Europe, you can see that quite often or not quite often, I believe there are no females that are actually affected by hemophilia because it is following this X-linked or sex-linked pattern of inheritance. All right, so sometimes the environment has an impact on your phenotype. So sometimes you have a genotype that doesn't match your phenotype because the environment is affecting the way that that phenotype is being expressed. So an example of this is a disorder called phenylketonuria, and if you are not affected by this or don't know somebody who is affected, you've probably never noticed that on the side of a can of Diet Coke, there is this disclaimer that says phenylketonurics contains phenylalanine. So this is kind of like an allergy warning. Now this condition results from the inability to convert the amino acid phenylalanine into tyrosine. So this disease is an inability to perform a metabolic activity. Now, people who don't have this disorder are able to convert just fine, and phenylalanine does not accumulate in the body. And if it is converted into tyrosine, then phenylalanine and tyrosine can both be used to build other proteins. But the problem is that at birth, some people carry 2 copies of the mutant version. So this is a recessive allele because we require 2 copies. And the recessive allele is supposed to code for this enzyme that converts phenylalanine into tyrosine. So an individual who is homozygous recessive for this has a complete inability to do this conversion. So little by little, individuals consume phenylalanine and it builds up in their bodies. So within a few years, this accumulation of phenylalanine can reach toxic levels and basically poisons the brain. It can lead to mental disability and some other health problems. Now, the reason that we say that this is affected by the environment is that by limiting the amount of phenylalanine that is consumed, individuals who are homozygous recessive can actually avoid this sort of toxic buildup of phenylalanine in their brain. So if they're managing it correctly, even though their genotype says that they have phenylketonuria, they may be able to avoid the disease. So we can sort of think about our genotype as really kind of a almost like a loose guideline for what our phenotype is going to be. So I tend to think about DNA as kind of a blueprint. It's the instructions for building a functional individual. But really, because the environment can have a huge effect on our phenotype, it's really more of a guideline. It's not so much a blueprint. And that's because we've got environmental effects, we've got additive effects, we've got polygenic inheritance, we've got all of these things that can affect our phenotype, even though our genotype tells us something differently. So another example of the environment affecting phenotype are the pigmentation in these Siamese cats, rabbits. This happens quite a bit in different organisms that have dark pigment. These Siamese cats and this Himalayan rabbit carry genes that produce dark pigmentation. So these genes are actually very affected by the environment and are actually heat sensitive. So dark pigment is produced only in relatively cold areas of the body, while warm areas remain really light. So this is why fur on the coldest part of the body, the ears, the paws, the tail, the tip of the face, are the darkest, while the fur on the rest of the body remains light colored, cream colored, or white. So it's interesting that Siamese cats that live in cold climate and spend a lot of time outside have significantly darker color in colder winter months because their skin is colder. Those that are indoor more often in winter will remain a little bit lighter in color. So you can see here we've got the areas that are cold. Think about when you get cold, your ears get cold, your nose gets cold. So we see the darker pigment there. The rest of the body that tends to stay warmer, your core, the rest of your head, are going to be lighter. So there are thousands of other cases in which genes interact with the environment and influence the effect on the body. The range of environmental influence traits are what am I trying to say? Help to sort of affect some of the other continuous variation that we see in humans. So an example of this is body weight and caloric intake. So you might have the genetic predisposition to have a smaller frame, but if you have too much, you consume too many calories, you're going to have a larger body frame, larger body size. So there are, you can also maybe make an argument that something like intelligence has an environmental characteristic. Being in a situation where you can learn and thrive is going to basically make an additive effect on top of your genotype. So there are a lot of traits that are influenced by the environment. All right, so I apologize for switching gears again. Our book is hopping around a little bit. I thought about putting this section back with our stuff on Mendel, but I decided to just leave it where it was for the sake of continuity with our book. So we're going to go back to Mendel, and the reason that they have this section here is that we are going to talk about the law of independent assortment, which discusses how two different genes can be inherited together. So because we're kind of talking about multiple genes, multiple alleles, they put it here. So the law of independent assortment basically says that the probability of receiving any particular allele for one gene on a chromosome does not affect the probability of receiving a particular allele on another gene on another chromosome. So I think this is best illustrated by an example. So here our book uses the example of short hair and white fur. So the law of independent assortment would say that the chance of inheriting the short fur allele does not affect the chance of inheriting the white fur allele. So we are going to do something called a dihybrid cross here. In fact, I'm going to move forward just a minute and then I'll come back. In a dihybrid cross, you are crossing two individuals who are heterozygous for two traits. And if they are following the law of independent assortment, you have an equal chance of receiving either of the allele combinations that are possible. So just like the other examples, I will do some examples in class because I feel that it's easier to see on paper when you actually do it. But just like our other Punnett squares, we're going to start out with a table of gametes. So if both of our parents are big L, little l, big W, little w, we are going to make that cross. Now you start by making all the possible combinations of gametes that that parent could make. So if the mother has short hair and white fur, but is heterozygous, she is big L, big W, little w. So her gamete possibilities are big L with big W, big L with little w, or the opposite, little l with big W, little l with little w. So that's what we have in our gametes up here. Same thing for dad. He's got all the same gamete possibilities because his genotype is the same. So then we do our little pseudo multiplication table here, and we end up with big L, big W, big W. So this is an individual who has short fur and white fur. Over here, big L, heterozygous for W, still white with short fur. Down here, we have a typo in our figure. This should be a little w because we've got big L, little w, big L, little w, just like this guy over here, little w, little w. So we go through this table and we basically, I call it multiplication. You multiply them together, you get big L, big W, etc. You get your L's and your W combinations. Down here, we start to see the long hair. So here, the recessive situation is little l. That produces long hair. This individual is white. This individual is yellow. This one down here, this one little guy is homozygous recessive for everything. So he is short-haired and yellow. So over here, I'm actually not sure what's going on because we have white fur. This must be a different example. Sorry, I don't know why they're doing a summary over here because this is saying pigmented and albino. Anyway, let's ignore this over here for now. Basically, what you would do with this chart here is you would tally up, and if you tally up, you will see that there are 9 individuals that are white and short-haired, 3 individuals who are short-haired and yellow, 3 who are long-haired and white, and 1 who is long-haired and yellow. So I have gone back and fixed the chart and figured out what's going on here. I was confused because when I read the book it made sense, and when I looked at the chart on the PowerPoint it did not make sense. So anyway, let me explain what's going on this chart on the right hand side. So basically this chart is saying that these events are individual events. So if you were to just look at the fur, the short hair versus long hair, you would get 3 out of 4 individuals being short-haired, 1 out of 4 individuals being long-haired. And if you were to tally these up, that is what you would see. If you look at fur color individually, 3/4 are going to be white furred and 1/4 are going to be non-white. I don't know why they say non-white. They should say yellow because that's what they're showing. But anyway, if you were to multiply these probabilities together, you actually can find the probability of being both, say, short-haired and white furred. So the point of this is to show you that each of these are independent events, that the probability of being of having a particular hair length has nothing to do with the probability of having a certain fur color, but you can determine the overall probabilities using that multiplication rule that we used when we were looking at pedigrees and Punnett squares. So last thing that we're going to talk about is the idea of linked genes. So we've been talking about individual genes, or we've been talking about genes on 2 different chromosomes. That is independent assortment. But if you think about it, we have a lot of information on any particular chromosome. There could be hundreds to thousands of genes on any one chromosome. And that means that sometimes traits are inherited together more frequently. So if you were to do a dihybrid cross, you would expect to follow that 9:3 to 3:1 ratio. But sometimes genes are close together, and when they are close together and inherited together in high frequency, we say that they are linked. So a really common example of this is red hair and freckles. So very often individuals that have one of these traits also has the other, and this is because they're found closely together on the same chromosome. So we can see why when we go back to the process of crossing over. Now remember, when we were in meiosis, we had this crossing over event that exchanges genetic information between two chromosomes, and the purpose is to hold them together in a tetrad to get to the metaphase plate. Now, when you have linked genes, they are likely to stay together in those crossover events. Now, I'll let you look at the differences between the blue chromosomes at the top and the different combinations. But basically, if we are looking at this as red hair and freckles, the dark colored genes, these guys are linked together. So even when crossing over occurs, they stay together. Now these are, we can also see the blue up here, those are genes that are not linked. They're not positioned closely on the chromosome. And so we can get those in different combinations with the red hair and freckles, but the red hair and freckles tend to stay together. And we see this sort of bundling effect when two genes are located closely together on the same chromosome.